People engaged in prolonged fasting consistently report a strange conflict. They sleep less and wake up multiple times in the night, yet report feeling hyper-alert the next morning, all while their bodies undergo accelerated cellular repair. This occurs because the brain's evolutionary survival drive is actively competing against the homeostatic need for rest. To understand how these two systems interact, we have to look closely at the structural architecture of sleep itself. This hypnogram charts a healthy, fed sleep cycle over an 8-hour period. You can see the distinct consolidated blocks of REM sleep and the deep, restorative N3 slow-wave sleep. When we overlay the data from a subject on a strict 48-hour water fast, the architecture shifts. The REM blocks shrink dramatically. In the deep sleep stages, we see extreme fragmentation. These red spikes indicate wake after sleep onset, or WASO, moments where the subject snaps from deep sleep to full wakefulness. Finally, the entire sleep timeline is truncated, often losing 1 to 2 hours of total sleep compared to baseline. Standard clinical analysis would classify this specific architecture as severely degraded and harmful to the subject's recovery. Nutrient scarcity restructures sleep by triggering a systemic survival response. However, by mapping this chemical cascade, we can isolate the cellular repair benefits while maintaining restorative rest. The origin of this sleep restructuring lies in a specific neurological pathway. The lateral hypothalamus acts as the central control room for the body's response to nutrient deficit. As blood glucose drops and the hunger hormone ghrelin rises, these signals converge on this specific node. This chemical signature activates a cluster of orexin neurons. Evolutionarily, this is known as the foraging response. A mammal deprived of nutrients prioritizes alertness over sleep. The emergenic pulse maps outward from the brain down through the spinal cord, increasing the overall tone of the sympathetic nervous system. This increases the release of adrenaline and noradrenaline into the bloodstream to mobilize stored energy. Through lipolysis, the body breaks down stored fat, expelling lipid droplets into the system to fuel the demands of the foraging response. This chemical cocktail produces a hyper-clear, wired state of mind, a sensation of high energy despite the lack of caloric intake. This systemic arousal is a calculated biological survival program that overpowers the homeostatic sleep drive. We can track the exact impact of this orexin surge by measuring the resulting shifts in the resting brain. Looking back at the REM stages on the fasted hypnogram, the suppression is significant. This occurs because REM sleep is metabolically expensive. During REM, the brain consumes nearly as much glucose and oxygen as it does when fully awake. To conserve energy, the central nervous system aggressively suppresses REM cycles. In contrast, deep, slow-wave sleep remains stable to enforce physical energy conservation. However, the orexin drive forces extreme fragmentation, leading to frequent WASO awakenings. This manifests as waking up multiple times a night and snapping instantly to full alertness rather than feeling groggy. Eventually, the high state of arousal simply truncates the night, reducing total sleep time without inducing the expected daytime fatigue. The entire fasted sleep architecture is a highly orchestrated adaptation, masterfully balancing deep physical energy conservation with absolute survival readiness. While fasting sleep is fragmented and high in sympathetic stress, the cellular environment during these periods undergoes a different form of maintenance. This split view shows the difference between a cell in a fed state and a cell in the energy-scarce environment of a fast. The lack of cellular energy acts as a severe biochemical trigger. Energy depletion heavily phosphorylates AMPK, the primary metabolic trigger for nighttime autophagy. AMPK activates FOXO3 pathways, which act as cellular sweeps, clearing out misfolded proteins and debris. While this occurs in the cytoplasm, a separate repair mechanism initiates within the cell nucleus. In a fed state, cellular NAD+ molecules are constantly consumed, but without dietary energy to manage, the cell maintains a high surplus of NAD+ throughout the night. This surplus allows SIRT2 and SIRT6 enzymes to move away from metabolic stress management. Instead, these powerful enzymes reallocate their entire bandwidth to actively repairing DNA strand damage and maintaining long-term genomic stability. This systemic energy crisis forces the specific biochemical environment required to run repair pathways at a significantly higher rate than during normal fed sleep. Harvesting these cellular repair benefits without the physical toll of disrupted sleep requires a specific intervention. The Fasting Mimicking Diet, or FMD, is a framework designed to act as a biological safety valve. FMD utilizes a low-leucine, plant-based macronutrient profile that provides trace amounts of complex carbohydrates and fats. These trace nutrients are designed to blunt the severe ghrelin and orexin spikes before they trigger full sympathetic arousal. If we look at this third hypnogram, we can see how the FMD alters the sleep architecture. The severe WASO spikes disappear, and continuity returns to the deep N3 sleep blocks. There is also a partial recovery of REM sleep, as the brain no longer needs to enforce extreme energy conservation. The nutrients blunt the brain's panic response, but the total calories remain low enough to maintain the state of systemic fasting. This low-calorie state ensures that the AMPK and sirtuin repair pathways remain fully engaged throughout the night. The FMD biochemically uncouples the stress of the foraging response from the metabolic benefits of energy scarcity. By understanding the neurochemical levers of starvation and arousal, clinical protocols can bypass evolutionary panic, allowing the brain to rest while the cells rebuild.